So now what we can do is basically select all those comparisons that are relevant to what we're looking at for a meta-analysis. And again, in this case, what we want to do is get comparisons that contain papillary renal cell carcinoma. All we do to get those comparisons, and again, all the comparisons are given here, and the number of genes that they found differentially expressed based on their criteria. All we have to do in order to collect that is basically click on that gene list. And I've already actually done that. I've collected the meta-analysis. But again, here you can see that we have 17 different comparisons that we're using. If I click on this gene list, now it goes up to 18. So now this gene list, these 12,561 genes are now part of my meta-analysis if I want to. I'm going to take that off. But basically that's as easy as it is just going through here, going through these studies based on our filtering criteria and finding those comparisons that are relevant to what we're looking at. And what's nice is what you're going to find is there's comparisons of papillary cell carcinomas to normal kidney and also different forms of the cancer compared to itself, which gives us a kind of variety here. So again, I just went through here and just picked all the data sets that I could that contain the condition that I'm interested in. We can then, once we've picked all these, now what we can do is go to meta-analysis, see those genes, and then get some of the biological information that's associated with that. So let's click on this icon here. It looks like a funnel here. So given in the setup are basically all the comparisons that we picked for our meta-analysis. Again, these are from 8 different studies. I mean, there are millions of dollars worth of research data in all these comparisons, and we get to use it, which is really, really cool. And the nice thing is we don't have to believe just one group. You know, we can take all the results and basically get our own information. The nice thing about this page is you can kind of put this wherever you want. You can move these up and down. So what I tried to do is kind of go from the most mild form of the cancer to the more severe. So kind of all types of cancers, type 1 and then type 2. And then the last 3 studies are basically different forms of the cancer compared to itself. So in this case here, it's type 2 cancer, but it's the hereditary versus the sporadic form. Here it's the second to last one is type 2 versus type 1, and given in the last one is poor survival versus excellent survival. So now that we have all these studies, now what we can do is say, okay, what genes are in all these different lists that are common? And that is given in the next tab right here. Load up, and here we are. Given below are some of the genes that were differentially expressed in our 17 different comparisons in our meta-analysis. What's nice is it gives you this nice kind of visual bar chart, so you can kind of see what your gene did in all your different comparisons. So if the bar is red, that means that gene was overexpressed in that particular study compared to the control. If the gene is green, that means that particular gene was underexpressed compared to the control. The height of the bar gives you the relative magnitude of that fold change. And what's nice about correlation engine is that the highest fold change in any study, they basically make 100. So all the different fold changes are basically ranked from 100 down to 1. So we've got a score now that will allow us to kind of compare all these different fold changes, given the fact that, you know, if I did a RNA-seq study, it tends to be a lot more sensitive than, say, an array study. So I'm going to get bigger fold changes, per se, with the RNA-seq study. This kind of allows us to kind of correlate all that stuff together. Also, so remember in our meta-analysis setup, most of these studies we're looking at the papillary renal cell cancer versus normal kidney. These last 3 we're looking at different forms of the cancer compared to itself, and what I find interesting is that for this particular gene, it is lower in the type 2 and the poor survival versus excellent survival. And then when you compare this gene, when you compare it to the normal tissue, it's all up. And that's kind of typical of what we find with a lot of these cancers, is that you might have a gene that's important to cancer for both, but it's actually maybe more important to some form of the cancer than the other. And we can just look down here. If you remember back in my my first presentation, I gave you a background on papillary renal cell carcinoma. I mentioned MET. This is the MET proto-oncogene. Again, if you go back, MET is actually tends to be more associated with type 1 than type 2, and sure enough, when we're actually looking at type 1 versus type 2, which is this column here, I think so, yep, we are seeing a decrease. And you can actually click on this bar and see the fold changes in p-values for all our different comparisons in our meta-analysis. So here for the MET gene, here are the p-values associated with each one of those different studies and the activity value, or in this case, fold change. So these are the genes that we're actually going to use for our meta-analysis. What we can also do is actually basically look for overrepresented biological associations of these genes, and that will be the next tab. So let's take a quick look at that. So go up here and click on Biogroup Results.